A-Level Biology 9700, Paper 5, Variant 2, February, March 2026. And we are going to discuss question number 2. And question number 2 is Daphnia magna is a small invertebrate that is found in many freshwater ecosystems. Figure 2.1 shows 1D magna as seen with the light microscope. The body of D magna is transparent and the beating of the heart can be easily observed. You can see the figure 2.1. So a student obtained a sample of pond water containing several D magna. To measure the time for one cardiac cycle in D magna, the student used a pipette to place one D magna in the center of a petri dish, added a small amount of cotton wool to reduce the movement of the D magna, added 2 drops of pond water to the D magna, observed the heart of the D magna using a light microscope, counted the number of cardiac cycles, heartbeats in 1 minute. In a try experiment at 20 degrees C, the student counted 232 cardiac cycles in 1 minute. So calculate the time taken for one cardiac cycle. Give your answer in seconds and two significant figures. So here we know that the student counted 232 cardiac cycles in 1 minute. So we have to find out the time for one cardiac cycle. So for this purpose, first of all, we are going to calculate this as total time is equal to 60 seconds, 1 minute. And total cycles are 232. So time for one cardiac cycle is 60 divided by 232. So it will be around 0.25862. So we will write the answer as up to two significant figures. So it will be as 0.26 seconds. 0.26 seconds. Now we are moving towards the next part of this question. And next part of this question is caffeine is a drug found in tea and coffee. The student decided to investigate the effect of caffeine concentration on the time taken for one cardiac cycle in D magna. The student was given a 0.5% stock solution of caffeine from which to prepare a range of caffeine solution of known concentration to use in this investigation. The student made 20 cubic centimeter of each solution. Describe a method the student would use to make a proportional dilution of the 0.5 stock solution of caffeine to obtain a 0.3% solution of caffeine. So first of all, we will go to find out the volume of stock solution needed for this purpose. So dilution equation, you know all. I will start with that. It is C1 V1 is equal to C2 V2. So here you can see the C1 is 0.5%. C1 is 0.5%, which is stock solution. And The required is 0.3. So C2 is 0.3%. So we have to calculate the volume of stock solution. So it is V1 has to be find out. So where they have given V2, which is 20 cubic centimeter. So this is the final volume. Now we have to calculate this. So according to this formula, we have to calculate the V1. So V1 will be equal to C2 V2 over C1. So C2 is 0.3 multiplied by volume is V2 is 20. So it will be divided at 0.5, which is equal to 12 cubic centimeter. So after calculation, I have written this. So we are moving towards that. Now volume of distilled water. So it is 12 cubic centimeter. To make a solution of 20 cubic centimeter, we need to add a volume of distilled water 8 cubic centimeter. So I will write over here volume of distilled water required is 8 cubic centimeter. So this is the calculation. Now I have to write the answer because we have to describe a method. So we have calculated this. So we will write is this. Measure 12 cubic centimeter of the 0.5% caffeine stock solution using a miring cylinder. Or pipette. You can also use the strange word syringe over here. Second, add 12 cubic centimeter of the distilled water. So this is 8 cubic centimeter of which we had calculated water to make a total volume of 20 cubic centimeter. Then mix thoroughly to obtain a 0.3% caffeine solution. Next part of this question is describe a method that the student could use to investigate the effect of caffeine concentration on the time taken for one cardiac cycle in D magna. Your method should be set out in a logical order and can be detailed enough to allow another person to follow it. You can assume that a detailed Risk assessment has been prepared. Details of how to carry out a proportional dilution should not be included. So here we have to write a method uh, which can be helpful for a person to follow. So I am going to write the details over here. First of all, always we have to write about the independent variable and then dependent variable and the all those points which should be which can be added into it. So first of all, I am going to write which can be the independent variable over here. So we will start with it. Prepare a range of at least 5 caffeine concentration concentrations using proportional or serial dilution. So it is in the independent variable. Now we will write about the dependent variable, which will be you can see uh, as place one Daphnia magna, Daphnia magna onto a petri dish with a small amount of cotton. Add a set volume of the selected caffeine solution using a pipette under light microscope and count the number of heartbeats in a fixed time. using a stopwatch. Then calculate the time for one cardiac cycle. The controlled variables temperature, should also be observed. Keep temperature constant by using heat filter or cold water bath. Home port or cold water bath or LED lamp. So they can be used for this purpose. Allow the dyspnea a set time in each caffeine solution to acclimate before taking measurements. Use dyspnea of similar size and age. Repeat the procedure. Repeat the procedure for at least 3 to 5 times. And then calculate mean time for each consideration at each consideration, and you can also find obviously anomalous results. So it was all about question number 2. Now students, please like, share and subscribe my channel to watch more videos on the coming recent papers. Thanks for watching.